Okay, awesome. Thanks all for joining. It's my privilege to welcome George to be presenting to us today. George and I met a couple years ago at a multiplex assay for variant effect workshop over in the UK, and have had a lot of really nice discussions moving forward regarding oncogenicity guidelines and modeling, MAVE experiments for testing for cancer oncogenicity, as well as he had some really nice feedback for us based off of the fusion work group. And George has just been a really nice contributor and a real pleasant person to work with. George is the deputy lead of Cancer Genomics Services at NWGLH over in England. And he'll be presenting the work from the SVIG UK work group on their guidelines for classification of oncogenicity of somatic variants. So take it away, George. Thanks for being here.
Thank you very much, Wes, and thank you for the very kind introduction. So can I just double check that you're still seeing the presented slides? Excellent. Thank you very much. So yes, as Wes said, I'll be going through some of the recommendations that we have put together in the UK. A lot of it will be familiar, and you're an expert group, so I will not be going through the details. And if you want to interrupt me, if you want to have any questions, please do that while I'm going through it if you like to. So again, I don't need to talk much about the importance of having guidelines, but we in the UK, and I'm sure it is the case around the world, there's a lot of variation when you come to interpreting somatic variants. So when we brought that group together, that was in 2018, and the aim was to try and establish a consensus, a tumor agnostic approach that we can use to standardize somatic variant interpretation. And we were wanting to separate biological significance interpretation from interpreting a clinical activity. And the reasons that we need this, as you all know, the ACMG guidelines are not designed for somatic variants. There have been few, when we started, there have been few interpretation frameworks available, nothing widely adopted. And then towards the few years into our work, of course, HORAC was published, which was a really good oncogenicity framework. We thought about whether we do need to carry on or whether we need to stop. And for that, we did a comparison between HORAC and the guidelines that the framework that we had already in place, developed at 2022. And we decided to press on with our guidance. And on the right hand side, just again, similar to HORAC, we've adopted from HORAC actually the oncogenic terminology, and we do provide the same scale that is based on the ACMG and the points-based system that we use here in the UK a lot as part of the cancer variant interpretation group, which is the germline equivalent of the SVIG. And this is not designed for actionability. It's designed to go hand in hand with the AMP tiering. So these are our evidence criteria. So we have 11 oncogenicity criteria, and we have 7 benign criteria. We have variable weighting for each code. That's one of the points that we have with HORAC is the number of variable weightings that are available within our guidance, which is also designed, we think it is a little bit more flexible, but also designed to align with the inherited or germline cancer variant interpretation group in the UK. We also have a permitted restricted code combination, again, aligning with the work that we have done in CanVig UK. So what I was going to do is I will go briefly through some of these points, and then, like I said, we can have questions or discussions. But just because I'm mentioning it many times, In case you don't know it, cancer variant interpretation group in the UK has been established in 2017 in order to align the ACMG guidelines. At the time, there wasn't many other follow-up publications to cancer variant interpretation, germline cancer variant interpretation. And we have a really nice a Canva and Canvig website where you can see a lot of information. So we put our consensus specifications on it. We also have various gene-specific recommendations, some functional study scoring, which is important for a lot of the big cancer genes, and also a Canva UK variant database. Again, this is currently mainly germline, but we are looking at options to have also a somatic database. I don't know if you use Cosmic much, but it is now gone behind a paywall. And as you have, as probably if any on the call are from outside the US, you may have experienced difficulties to accessing Genie recently. So we're trying to build a national somatic repository. It might be presented on the Canva website or somewhere else, but that's also work in progress. So our evidence criteria, one of the criteria that doesn't exist in ORAC, but is we adapted it from Fryen et al., which is the Belgian group, was this idea of canonical variance. We wanted this to be a systematic list, so we set some criteria for it. It has to be mainly oncogenic. There are a few exceptions, so a minimum 10 points, no discrepant criteria as well, and specifically being highly enriched in tumor databases, so which means O4 strong, which we'll see in a minute. We think this is useful because it reduces effort for labs, especially smaller labs, of having to go through some of the commonly encountered variants. If groups are starting new, then they have somewhere that they can start from. So this is just a snapshot of the list. It's a bigger list. Like I said, most of them are oncogenic. There are very few exceptions that only met likely oncogenic, but there are all enriched in somatic databases, and we didn't feel the need that labs need to be going over and recording these. So if the variant is on this list, they can call it O1 and they don't need to classify it any further. Then we have O2 and O2 RNA, which is essentially a null variant being present in tumor suppressor gene with a full flexibility between supporting and very strong. This is essentially PVS1 in the ACMG criteria, if you're familiar with that. There are specific recommendations for cancer-specific genes, especially when there are VCCs available with specific criteria, like the Enigma for BRCA1 and BRCA2, and the MMR genes with the INSIGHT criteria. The benign equivalent of those, not always fully benign equivalent, but the benign side of this is when the variant does not fit the mode of action of the gene under investigation. So, for example, loss of function variant in an oncogene, you can directly put that as a variant of uncertain significance without having to do more work. Again, this is trying to help labs find workflows that will speed up the process. I do work on both germline and somatic, and when I work with labs that are purely somatic, a lot of them have not been used to a very detailed variant interpretation, and that's why we have been very conscious in trying to streamline the process as much as possible to reduce the impact of introducing guidelines. Labs that work with germline, at least in our experience in the UK, labs that have germline facility or is a combined germline somatic have had a longer standing experience in applying structured guidance for variant interpretation. There's also somatic or intronic variants with no prediction on splicing. Again, that can be strong. There's also the RNA element of that, so that's not just the in silico prediction, but the ones that have had studies similar to O2 RNA to show that there's no impact on the RNA. O3 is absent or very rare in population databases. Now, we know that in the new ACMG guidelines, the equivalent of this PM2, there will be no presence of PM2. There will be other things that will balance the books, but we agree that absence in databases is an evidence for rarity rather than evidence for pathogenicity. However, to align with other guidance in the UK, and while we're working on adjusting other parts of the guidance to balance missing this element, we have kept this criteria, but we have kept it more strict than what's used in the ACGS guidelines in the UK, which is the guidelines used for inherited variants in general, rare disease. So we kept it fully in line with the CanVIG UK. So that to achieve a moderate, it has to be absent in all population, including nomad version 4, which includes the, as many of you know, if all of you will know, the huge UK Biobank input. And then, so most of our variants are ending up with supporting because there are some occurrences. The equivalent, the benign equivalent of that, of course, is the variant being very commonly present in a population. If it's greater than 1%, it can be a standalone benign. Greater than 0.1%, it's a strong piece of evidence. We are thinking about making this even increasing the or lowering the levels that you can achieve standalone and strong, but we are looking at data in the UK before we do that. So we know that this is relatively strict, and we're hoping to make it less so. Enrichment in somatic databases. This is if the variants have been seen often in large data sets. So for example, Genie and or a national multi-center curated database. And like I said, we are working now on collecting data from, if you've heard of the 100,000 Genome Project that happened in the UK, plus lots of large data sets. We're pulling lots of this together to create a national resource that we can use. We think this is being on the permissive side in terms of allowing these numbers to be used at these levels. We're hoping to move, once we establish a national database, to move to having a record with denominator, which will allow us to apply some statistical tests for enrichment, similar to what happens on the germline side. But to start with, this is what we're suggesting, and we're doing various things for missense and splice, frame shift, and in-frame deletions. It is mainly tumor agnostic, or with the prerequisite that these needs to be a matching mode of actions. So if you are not confident that different tumor types have the same mode of action, we recommend that you strictly stick to the same cancer types, the same tumor type. There's also O5, which is variance. It previously used to be known missense variance at the same location, but we know it has been expanded many times beyond missense variance. So this is the equivalent germline of PM5. So again, we've adapted CanVIG germline guidance, which many of it has been on some of the recent VICC publications in this area. So we try and put as much details to allow the application of this in a systematic way. There's also O6, which is the in silico predictions. Essentially, Revel is the main scoring that we use. We're still using only supporting level here, so we haven't expanded to the Pjavr publications where you can use multiple levels. And again, that is to align in the UK with across somatic and germline. And there are many reasons why we have to do that, at least in the UK. And we think it's useful scientifically because of a lot of the overlap between somatic and germline testing now, whether genes that are can be tested in a somatic manner, but they have high likelihood of being germline in origin, like BRCA1, BRCA2, and MMRs. We do a lot of testing in tissue for diseased cases. And there's a lot of overlap now, expanding overlap between testing for treatment based on germline somatic. So there is a lot of overlap for us. We follow something called the test directory in the UK, where it standardizes what tests are available for what patients in the NHS, the National Health Service. And there is a quite good overlap between germline and somatic now in cancer testing that means that we're trying to keep the guidance also aligned to avoid confusion and errors. There is O7, which is located in a mutational hotspot or a well-established functional domain, again, in line with some of the germline guidance with the flexibility of using supporting moderate and strong cancer hotspots is the database that we highly recommend for finding these functional domains. O8 is limited at supporting. This is where there is the constraint for missense variation. Again, self-explanatory, and it's based on the Z-score available on NOMAD. O9 is where there is a protein length change in frame deletions and insertions. It varies in strength based on if it is a greater than one amino acid, limited to one full exon, or if it's a smaller, essentially affecting a single residue. The benign equivalent of that if the in-frame deletion or insertion happens in a repetitive region and there is an unknown function there. O10 with the B6 benign equivalent is essentially functional studies. PS3 if you use the germline, which based on the Berneck et al. Publication from 2020. Now this has caused a lot of issues with when we were doing the testing for the guidelines because again, many people found this very strict. If you have used PS3 or followed the Berneck recommendations, you know that a lot of the older functional studies struggle to achieve a good level of evidence. As presented previously in multiple papers by the Klingen SVI group, PS3, the functional study evidence, was one of the highly variably applied piece of evidence, which caused a lot of differences between labs. So I really like standardizing this. Yes, it is strict for older functional studies, but we do sometimes need to be more strict. We have come across a lot of variants where there have been some poorly structured functional studies that resulted in the wrong interpretation. And as was mentioned, we met in the one of the meetings for MAVES, which is the large functional effect studies. We're hoping that as more and more of these studies now move toward cancer genes that are important in a somatic setting, tumor setting, more of them will be achieving this higher scores of evidence. So it is strict. It is the point that many people did not or many labs that responded to our guidance consultations did not like, but we wanted to stick with it to improve standardization, consistency, and also increase the quality of the functional studies that we accept. O11 with the equivalent of P7 is one that doesn't exist to a similar effect in other guidelines. So this is trying to use all the tumor phenotype information in a way that can be used to give extra pieces of evidence. So there are some examples that we provide in the supplementary part of the guidelines, like using microsatellite instability, using HRD phenotype, loss of heterozygosity of the gene, or other immunohistochemistry analysis. That doesn't always reflect necessarily a variant-specific impact, but can help point in the right direction. This has been very well received in our consultation. Many labs really like having this piece of evidence. It allows us to use data that come from other testing that happens on the tumor. There's also the table of allowed and disallowed combinations. So this was initially done by our CanVIG group, and we always found this really helpful. We know that a lot of the data that we use in variant interpretation overlap between different criteria or different pieces of evidence. So it's really important to review the combinations. And what we did based on the CanVIG and with consideration of what is appropriate for the SVIG criteria, we reviewed all the O1 to O11 criteria across each other with any combination, and listed what we think is a not permitted combination, permitted combination, combinations that should be restricted. So you should be very careful when you're applying them, or apply them in certain circumstances, but not all circumstances, just to allow people to remember that, or to guide people to avoid double counting of evidence between different criteria. Same applies for the benign side. It is more, it's a smaller set of restrictions, much wider on the pathogenic or oncogenic side. I would highly recommend reviewing those, and also if you do feedback to us, that would be really helpful. So another important point in our guidance is this idea of overlap between somatic and germline. We do a lot of testing. I'm sure it happens in many other labs where you're testing a tumor material, you're reporting a germline variant and a somatic variant. And of course, there are guidance. I don't know if in the US you follow the same guidance. We follow the ESMO guidance on germline follow-up of tumor testing. And with a lot of some of these overlaps, for example, we do a lot of testing on tumors from Lynch cases where we're reporting the germline variant alongside a somatic variant. And we wanted to make sure that for the clinicians, the language is coherent. And the criteria, the classification that we're reporting back is coherent. So for a set of genes like BRCA1, BRCA2, and MMR, we have recommended, or any other genes that have expert curated criteria, guidelines, and have a high likelihood of being germline origin, we are recommending the use of the germline classification criteria and language. That does allow in these genes, for example, the tapping into evidence that would not be available for somatic testing, but also when you are reporting them in certain settings, it gives the clinicians, oncologists, clinical geneticists, or pathologists a more coherent reporting. From our clinical users, we had a really good feedback on trying to align between these guidelines in this way. It does create tensions on larger panels. For example, we do a lot of large panels for circulating tumor DNA, but we try to manage it slightly differently in the settings of the large panels. To also help the deployment of these guidelines in the UK, we have been developing a training package. So we have something called GTAC in the UK, which provides a lot of training and education, and we are creating a set of modules that allow scientists, newly qualified scientists, or scientists that are moving to the use of these guidelines to follow some tutorials, see some examples and tips alongside practice cases that to help in once labs adopt these guidelines formally, to allow for that standardization to be supported with training material and examples and practice cases where they will also see the answers to make sure that labs are adopting it in the way that we think it should be applied. Essentially, we have been working on this for a long time. We believe that it's a reasonable set of guidance, provides a little bit more flexibility than other guidance that currently in use. It also works well with germline guidance, and also work well with the actionability guidance. We have not included structural and fusion, for example, at this stage. We're considering what to do with that. I'm considering with Wes how we can bring some of this work together, which will also help international standardization in a better way. We have gone through the final stage of ratification by ACGS, which is the Association of Clinical Genomics Science in the UK, and submitting for publication as we speak. And there has been lots of effort from many people over the years. These are the main authors, or the core of the SVIG, but there has been a lot of input from many over the years. And that's it from me. Thank you.
Thanks, George.
Shall I stop sharing?
It was a nice presentation. Might have you keep it up in case people have specific questions. For instance, I was wondering if we could just roll back to 05 real quick. There was a lot on that slide.
Yes, it's yes.
Awesome. I was trying to take in the second image at the bottom.
Yes. Oh, sorry. Hold on. I'll bring it back. Oh, I've exited. I'll keep this for now, just so that I avoid having to exit again.
Yeah. Okay, so the bottom table is can you walk me through this again real quick?
Yes, of course. Can I check in terms of guidance? Have you used the Clinton SVI recommendations in this area? I'll bring the reference. Hold on with me, because this took a lot of mapping across. And what I wanted to say is what would be really good, and then depending on the others, we can follow it up either on this or offline. But essentially, have you used the Walker et al. Published in 2023? I'm just getting the reference now.
I'm not sure that I personally have.
Okay, so this is I would highly recommend Otherwise, we might be going in circles because trust me, we have gone through this with many of the users. Is I would recommend that you refer to this. Can you see
Mm-hmm.
my the paper? So this is what was published by the Clinician SVI group on the splicing. And what essentially it did is it expanded or clarified the definition of what you would take as a splice site, okay, and then provided a better alignment. So that's why it expanded beyond missenses to include many other things of when you can combine a variant under investigation or a variant under assessment, which referred to as VUA, alongside other variants, taking into consideration their predictions and taking into consideration the known impact. So let me just bring their lovely table, which we based a lot of this work on
Thank you.
alongside obviously referencing them. So what I would recommend to help make this an informed discussion is to, I'll share the link for this paper now on the chat, but the Walker et al. Really nicely expand the use of PM 5 and PS1 and the PP3 and so on, which allows you to use the splicing evidence in a way more informative manner than what we had available before this publication. So
Yeah.
I will put this link in because then it makes this table way more a better way to digest that table than what I would explain just now.
Yeah, no, I appreciate that. And I do appreciate your work group's thoughtfulness in utilizing work done by other groups to help standardize some of these. I do really appreciate that a lot. Another comment that I had just in general with the functional studies is I think I'm in full agreement that I would much rather be conservative in general with a classification, and really make it so that it needs to have good criteria to get it out of the VUS into likely oncogenic or oncogenic range, than accidentally have VUSs start bumping up into likely oncogenic.
Yeah,
That makes our clinical team obviously a lot more nervous. And so I think it's better to be conservative in the way that we apply these. And I do agree that the quality of functional studies is only going to continue to improve over time.
yeah. This was the one of the ones that was highly resisted in our community consultation. And over many meetings, we had to stand by. We really need to increase of our variant interpretation. Yes, it will take longer to start with, but it's also a training matter of making sure that people are buying into these criteria, understanding the reasoning behind it, because putting a value of very strong or strong, this is a statistical value or semi-quantitative value that we are putting in the points-based system. And for that, it needs to be supported by a structured analysis to align with that. So I'm happy that you're in agreement on that front.
Other questions or thoughts for George?
Yeah, this is Beth. I'm just wondering, you said that you've been involved with discussing MAVES. Where do you feel MAVES belong in our functional studies? Or how strong would you rate like a MAVES study right now?
So most of them do come up at strong. Um, but I think the interesting thing, and I work very closely with someone at the Sanger Institute that is designing a lot of these MAVE studies, is they, in many ways, sometimes jump over the question of oncogenicity to actionability. So a lot of these MAVE studies are not, some of them actually are not answering the question whether do you think something oncogenic or not. They're actually jumping straight away to response to treatment in a MAVE study style. And I think none of the criteria, none of the guidelines that we have nowadays cover that element in a good way. So some of the studies that I've seen are not, and like I said, the ones that I've seen for oncogenicity are excellent, and they do all meet the criteria of strong, but some of them are jumping the question of whether this is oncogenic or how this is oncogenic or what is the biological impact, and they're going straight into impact on treatment. And I don't think our criteria have a space for that at the moment. And this is something that we do need to work on allowing a space for in the future. But then it will cross between oncogenicity guidelines and the AMP tiering. So it would be a very interesting place to try and solve that once it's published.
That makes sense. Do you think that jump is misleading sometimes, because some of those variants that may be activating may just be resistant as well? So do you think that like making that jump to like therapy resistant or therapy response might be like kind of misleading, or do you think that the MAVE studies are generally, I just have limited exposure to them, but I'm wondering, do you feel like it's communicated well enough where you can easily, discern between different things? I don't know if that makes sense.
No, I think it does. I think yes, telling the difference between the resistance ones and that's really important. But I think what as a scientist, what I find sometimes worrying is giving that clinical label before we were able to work out what's the biological impact of that variant. But I think a lot of these studies are moving towards that way because it's giving them a higher impact publication. They are not trying to decipher the biology of these variants and their impact. They're directly saying whether a variant has an impact on treatment or not. And I think it's echoing some of the old ways of reporting somatic variants, because in the past you find a somatic variant in a patient with a tumor in a gene that you think or in a gene that has specific impact on specific treatment, and it gets reported that way. So I think it's echoing some of that in the past, but it might be the efficient way, and we just need to learn how to work with it appropriately, because they're directly answering the question, is this really going to impact treatment or not?
And I do think another important progress that the MAVE group is making is they're working on standardizing the data model that's expected for introducing data into MAVE-DB, right? And so Alan Rubin has been working a lot with the Global Alliance for Genomics and Health to standardize all of that, because kind of in a similar way to how some of older functional studies now we kind of consider like the Wild West, some of the earlier MAVE studies are absolutely the Wild West in terms of what they give you to be able to actually assess what the study, how it was done, all of that. And so Alan has worked really hard on standardizing what is expected within that data when people submit that, so that you can actually then do a more rigorous clinical interrogation of that data and whether you'd want to use it or not.
Yeah, I would just add that I think the MAVE studies, while really useful, are not one size fits all. Like you can't apply the same standard to every single MAVE study because all of the assays are different. And all of the assays, some of the studies are really well validated against known variants and some of them are not. And so I think it really does still require, I haven't looked closely at MaveDB recently, so I'm sure that the work he's doing is really excellent. And I think that the work that I assume he's doing is to assess each study individually and try to understand how strong the data is and how much you can rely on an individual call, whether
Um,
it's for biological relevance or ultimately clinical relevance.
um, so just to jump in here on the point by Shelley, yes, totally. I was giving a label in general that some of these MAVE studies are achieving strong, but yes, all of them will need assessing on individual basis following the PS3 guidelines essentially. So in CAMVIG as well, we have established a system where we assess functional studies, these functional studies, and then two competent scientists do the assessment, and we put the scoring on here. So it's something, if you go to the CAMVIG website and press on the functional study scorings, it will take you to an Excel that gets updated whenever large study has been assessed and reviewed, and we put the scoring that we think it should be used at. That's still mainly driven for tumor suppressor genes that have a high germline impact, but we're hoping with more somatic functional studies coming on board, that we will start uploading also assessments of these. And also a lot of the studies now are publishing with an assessment that they do themselves using the number of controls that they have validated against to say, we think you could use our study at, for example, PS3 strong, PS3 moderate, and so on.
Brendan, you had your hand raised for a second. Did you want to say anything, or are you good now?
It was thank you, Wes. My question was literally going to be, George, to ask you to talk about what your group's process is like to filling out this functional scoring, study scoring spreadsheet, and what that's like, but you ended up bringing it up in your answer to the last question. So thank you very much.
Thanks.
I do have another question. I'm sorry if I keep stealing all the questions. So as someone who our group is now actually implementing the 2022 VICC ClinGen CGC guidelines with modifications as well. So George and I have had quite a few conversations about this. George, I know that you guys had a large laboratory survey of actually like trying to implement this. How has the general reception of these guidelines been for each of your labs? And what ways have you all worked on trying to make the actual like implementation of these within workflows less burdensome and as less cumbersome on your clinical groups, because it is basically, introducing a whole new kind of workflow for them? What ways have you kind of implemented trying to standardize and help them to achieve this in maybe a programmatic manner or what? Just curious.
So thanks, Wes. So one of them is the O1. We also try to order the criteria with the ones that are more commonly used with more higher impact usually. We did add a point in the criteria, which was, I have to say, I wasn't a fan of it, but in agreement with the group, we added it, which is once you hit a classification, you stop. You don't need to continue. In the germline world, we don't usually approve of that because that's how you miss a contradictory criteria. Less often in a somatic world, but we are recommending to stop when you reach a classification and not worry about going through each pieces of evidence. Again, that's not the way we commonly done in the germline setting because we recommend that you review all of them. We also are putting together a stepwise framework that hopefully will help automate many of these steps. So we talked about O1, we talked about the ordering of the criteria, and we talked about stopping. So these are some of the initial steps, and also providing educational material to go along sided, and we're making an Excel sheet, protected Excel sheet that can be used to apply the guidance. It's still not fully validated, but we're also going to share that with all the groups that want to apply SVIG.
That's great. So then within your own labs, then is this information then being stored in a pretty structured way so that it can be reused, or how is that? I know that you just mentioned Excel sheet, which is useful, but then also makes me cringe internally because getting that data out of the Excel and then into like a structured database is a pain. What ways are you kind of trying to think about the implementation of standardizing this so you can reuse these, especially with your system? I would assume you can then reuse from another lab because they've gone through the same validation. How are you trying to make that happen?
So that's an excellent way. So I've mentioned a couple of times this national database that we're trying to work on. So this is a work beyond the SVIG group. SVIG group is part of this alongside Genomics England, which is the kind of the body that looks after a lot of the genome testing in the UK. And what we're trying to do is, because most of the genomic services in the UK happen under the National Health Service, we have a body called NDRS, which is the National Data Repository Service, that have rights by law to access a lot of this data. So what we're trying to do now for the somatic data, which we have been successful in doing for germline data for a while now, is to submit the data regularly to NDRS so that it's then pulled into a national database that can be accessed by everyone. It's a very early stage. It will help create a tool that's available for all the labs rather than all the labs trying to access different variant databases or submitting it to different or using different DSSs and so on. It's very early work, but we're hopeful that will mean, again, streamlining the process, mean that if other labs have done it, have seen it, of course, there are always limitations because you can't put the responsibility on someone else. You have to do a lot of things, but at least it will provide you with multiple sets of data and pieces of evidence that you can then evaluate yourself in a much faster manner, but still early on.
Cool. That's awesome to hear. I think, for me, always thinking about the ability to reuse data from trusted sources, right? So obviously with everything with the NHS, you're able to kind of do that because you're following a similar protocol. And so I think that's great. Other questions or thoughts for George? And George, these are up and available, right? At least preprint, if not all the way full through, right?
So they're available on the link that I shared with you, Wes, so please feel free to share that with the group. We have attempted putting them on medRxiv, but I don't know if many of you have dealt with medRxiv when it comes to guidelines. They have a very specific criteria on allowing guidelines to be published on medRxiv. They asked for very few changes. I haven't managed to do them yet because I was away for Easter, but what we're hoping to do is just to submit straight for publication. And we have a journal that we deal a lot with in the ACGS and BSGM, which is the British Society for Genomic Medicine, which is the Journal of Medical Genetics in the UK. We publish a lot of guidelines there. So we're going straight there for publication. They may be available through medRxiv before then. If not, they will be put on the ACGS website and possibly on CanVig as well. And thanks, Beth, for sharing the CanVig training resources. It's a widely used tool. There is a bit of it on functional studies assessment as well.
Mm-hmm. Yeah, I've been kind of poking around in there while we're talking and asking questions, and it looks like a really great resource just to get familiar with guidelines and things. So for those of us who do some training, we might be able to borrow from here.
And if you need any of the original resources, if you let me know, we can send you any PowerPoints or anything that might be helpful for you.
Great. Thank you so much. This was so cool.
Awesome. Well, this has been a really great presentation, George. Thank you so much for sharing all your hard work that your group has done here. I will make sure that this video gets uploaded as well as share that link out with the VICC for the publication. Anything else, George, that you want to mention before we close?
No, thank you. Thank you for the opportunity to speak here, and I'm looking forward, hopefully, to establishing more collaborations between us in the UK and yourself. So we'll keep the conversation going.
Yeah, absolutely. I think as we continue to move forward, the more that we can work together as a global community on these kind of things, the better it's going to impact all of us. So we really appreciate your hard work
Certainly.
and you presenting.
Thank you.
All right. Well, thank you so much, everyone. I hope you have a wonderful rest of your day.